But if you have no idea, it'll be too late to do everything over again. Okay, everybody take their card. So these are the people that, this isn't here. Brandon's not here. Sorry. Okay, so Alexander's not here. There you are. Okay, so I think it's 6 o'clock. It's really lecture time, but I thought we could, as we're here together, we could go through this. So if you go to Canvas, this is the best place to start. If you look at this, this is week 9, and this is when we were doing the phage. So on the bottom of the phage one, Thursday of that same week is when we started our project. So the first thing you should have read already is this portion about your unknown project. So this part right here is just telling you a little bit about that we did 2 plates. We did a gram stain and a quadrant streak plate, and we did it with our unknown for our project because we were going to use the TSA plate for something, and then we were going to use the gram stain to determine that you had the right organism, or at least you had the right morphology that you could look at. All right, so that's kind of what this is doing, and then this part up here tells you a little bit about your unknown project. Oh, maybe not.
Is that under modules?
This is under modules, yes. So are you under modules? Yeah, this thing does not open. I'm not sure they've been having problems. I tried it 2 or 3 times, and then it didn't work, and then it finally did. So it's just kind of trying it over and over again. So the module that we're going is week 16. So I open that up first. That's where we have the phage display, and then right underneath that is Thursday when we start our unknown project. So let me try it again. No, still not going to allow me to open it. Maybe if I wait. Anybody else able to open it? No?
No. It says it does not exist.
I know. It did earlier today. It was easy to open, and now it's not. So we might just have to go to something else. Let's see. So we'll try one more time. You can get all of this information in another place too, but it's just nice to start. No, it's not going. Okay, so let's just abandon that. So then we start where we're actually starting our unknown project. So here we should have our results table. Unfortunately, that first page that we would have would, maybe in here, requirements. Okay. So let me go back to show you where I went. In this week number, unknown project, so it's part of week 10, I guess. It starts out with this unknown results table, but you don't really know what that is and how it fits into your project. But when you go down to requirements for the unknown report, if you click on this, and then it gives you, kind of a description of what are the parts of the project. So you can't see a whole bunch here, but you can see that the project has several parts. There's something called 40. It's a media study. And it tells you exactly what you're going to do, and you're going to do it as a group, so that group is by table. So your table will do it. And you have a template, and I'll try to open up the template, but we can do that in a second. So that is going to be worth 40 points, and I don't have, let's see, I think I logged all these in to tell you exactly when they're going to be due. Oh, and I brought last year's so we can see a little bit about. Okay, so the top one where it says media study, that is going to be due on May 14th. That is not very far away. So May 14th. And let me show you, I think I might have an old media study in here. Okay, I have some.
It actually says it's due on the 13th.
It's due on the
Under assignments. Media study due.
oh, it is 5/13, so sorry. Yeah, 5/13. So here is a media study. So it's really hard to see, but this is from last semester. So they went through and they just filled out all the top things and they wrote everything down about all the organisms. And so it's going to look like this. So they had lots and lots of work put into it. So the media study is not a real easy thing to do. So make sure you kind of start early on it. And I think they give you some kind of starter material to do it.
Where's the template?
I think the template is, I think it's further down. So we're just looking at this. This is the overall document right now, and then there'll be individual ones. I think at the top it said media. Before we even got to here, let me go back and show you. So here's the project. And then here it says, oh, unknown expected, that's the word, the unexpected one, or expected. Here's a media study table down here. You see it up on the screen? Or you see it on yours also?
Yeah.
Okay. But we'll just go back to this to make sure that we kind of see the report and what's going to be required. So media study, and you can see that's a little bit further down, and you can open that up and look at it. It's worth 40 points, that's a lot. So you kind of imply that's a lot of work. It is per table, so every table will do this. And then you have the results table, that's going to be worth 25 points. And I can kind of show you what a results table looks like.
Are we, sorry, for the media study guide, are we turning in one per table? Or?
One per, well, you, every individual turns in the same one that on that table. Because I grade you all individually, and I don't want to have one document and then grade each individual person. I want everyone to hand back. So everybody will print it out, because you all contributed to it, and then everybody will get the same grade on it. Yep. Okay, so let me show you here's just a person. Here's kind of what, and there's some of these I think in the lab also. So here they have their organism, or characteristics of their organism. They had a certain unknown number, and then, and in fact that's what their unknown was. And then this is all the stuff that they did. Okay, so you'll have 3 columns to fill out. They give you a little starter, but it's, not a lot. Now, I don't like this one necessarily because I don't like just positive and negative. If it is something that turned dark in color, that was your positive exam, then I want you to type it on here. It had a dark color. Or if it's bubbles, it had a lot of bubbles or hardly any bubbles, or no bubbles, not just negative or positive. Okay, so really I want to see what your experiment did. So this will have a little bit more in here, a little bit, maybe 8 words or something under each one of them. Yes?
Is this something that we're going to start documenting our findings on like tomorrow?
You do it from day one. See, you should have already read all this. Like this was already last Thursday that you should have read it. And so to Tuesday that we've already had, and that has its section to read. So yeah, that's why I was kind of surprised to think, first of all, I said, have you looked at this? And he goes, no, I didn't know what that is. And I go, oh my goodness, okay. So I kind of showed him and I said, we better get started on this. But I figured that when everybody opened up the first day, you would have already seen the project and what you were supposed to be doing. But yeah, we need to start documenting everything now. And then one of the things I told you is make sure you take pictures of everything. because you will also have to do a presentation at the end of this. Now, the presentation is something that I do to give us it's an assessment, but it's non-assessment. It's not a quiz or an exam points. They're a presentation, and almost everybody does well on a presentation. So other classes do a lab practicum, or they do something that's another assessment. And I just didn't want to put more tests into our schedule. So I did this, and I think it's worth, I forget how many points it's worth. On the schedule, it tells how many points. It's like 25 or something like that. So everybody will do a presentation on their unknown. And it's not difficult to do because you've been doing this, the whole project, the entire time for that 3 weeks or so, 4 weeks. Yes?
And is there also a template for the presentation?
There's a rubric that I do for grading, yes, and so you'll get that one.
Are they going to notice like what we need to include?
It's pretty much a presentation that says you're giving it to an audience and you're explaining things. So I don't say, you must include this and this and this. It's your presentation.
So we just like describe the tests that we did and the results that we had that got us to our finding and what our unknown was?
Yeah, and way you went, maybe you went wrong, or the test that was the best test that led you to what you had to get. And then interesting things you found about it. Does it produce an antibiotic? Where is it found? I mean, so there'll be all your history that you found out about your organism.
Okay.
Something that you want to tell an audience. And you'll have, I don't know, I think we did 10 minutes per person last time, and they thought that was too short, but I think we have more people, so we may not be able to do it that many minutes.
Okay.
Anyway, so let's just go through this, and then as you start getting more and more questions, a lot of this is answered in the stuff they have in the manual. All right, so this is your media study guide. Also, we found there is a media study kind of a worksheet that you do at the Star Center. So last semester, students told me that was extremely helpful. It had almost everything that was on our list. It had it that they already, organized it for you and kind of told you what you had to do. So that's a good one to do if you have still room to do that. All right, so media study guide, 40 points, results table. I kind of showed you what a results table is. They will also give you in here an example of a results table. I just like a little bit more detail and not just plus and minus. That doesn't really tell me much. And then the identification. Oh, and here's their little sample of what a results table looks like. And then the identification of a logic tree, we'll talk about that a little bit more detail. But there are samples of a logic tree up on the bulletin board in the lab. So there's a couple of different unknowns, and they just have unknown number. I don't know what numbers they are, whether they're numbers that we still use or old numbers. And then they just show you how a logic tree can be done. And then there's, I think, a couple videos that they show you on how to do a logic tree. Okay, and then things like spelling, italicized, things like that. And this logic tree is going to be worth 20 points. And then we have a paragraph, and you can see the writing element of this is not very much. It's just kind of a, 3/4 of the page, half a page, or something like that. And this is only worth 10 points. So it's not a huge part of the paper, but it's good to start putting together, because when you do your presentation, this is where you're going to get all your information from your presentation. And then it talks about the references, making sure you have all the references in there. So here's a little description about the references. So in the references themselves will be worth 5 points. Yes?
When we did a presentation, are we allowed to have like notes with us?
Oh, yes,
or read
definitely.
while we're doing it?
Definitely, but you'll find out that last semester too was really nervous about it. But I said, yes, take 3x5 cards. And then no one used them, because you know your project so well. You've done all the information when you've written up your report. You've done your results. You've done everything, so you already know it. And you have slides that have everything that you need. So you're usually just kind of looking at the slide. Something on the slide is keying you to say something, and then you just talk. But yes, if you need note cards, that just makes you feel more comfortable, you can definitely have note cards. The only thing I do, and I will stop somebody if they start reading, is sometimes you're way up from your note cards, and then all of a sudden you read your note card and you go, oh, this is kind of easy, and then you just read and read and read and read. And then we're looking at you going, okay, talk to us. So if that happens, usually I just say, can you tell us more about what's on the screen or something? And that usually gets somebody away from the note cards. But yes, you definitely can. Okay, so that's your report in a nutshell. And then I will give you a rubric that will tell you what you're going to be graded on for your presentation. All right, so I'm not going to read all this to you, but you can read this. So this is pretty detailed. This is where you'll get all your information about how to do this. Then, I think on the bottom of this one, let me go out of here, they had the media. So this was the project. So up here was your unknown results table. So here's your results that you're going to have. Here's all the different organisms, very small, but here's the results that you will have. So you'll only have this column, and then you'll have your organism. Okay, kind of like I showed you, got 3 columns, but this is what I really suggest that everybody put together as a table, so that you start getting an idea of what you have right now. Okay, so if you know that you have a certain organism and it starts following a certain pattern based on this completed table, it really will help you know if you need to do a test over again. But, it takes a lot of effort and time to do this. So this is your results table. Okay, so let me go back here. And then I think the media was also on this page on the bottom. Here's your media study table. So that's the thing I showed you up here. So your media study table. And again, I believe that they have something very similar to this at the Star Center. So you might want to kill 2 birds with one stone, get some credit for that. Okay, so these are all the tests that we're using, kind of like the thing that we filled out up on the board for the gram positive. All right, so.
Sorry, for the media test study table?
This one right here?
Yes, you're filling that out for your own organism?
Yes. But if you have this other one done that we can go back to again, if you've already done this one with all the tests right here, if you've already divided it up and over a weekend or something like that, you just get it all done and you all share it, and then you start checking and go, oh, mine's really looking like this, and you say, but it also looks like this. Or it looks like this. So now you can start narrowing in. So you do another test and you go, oh wait, that eliminated that. And then you do another test and you go, oh, I'm not sure now because I have some different results. But at least you can kind of follow along. But if you wait to do this to the last minute, which you still have to do, then and then you're not really getting to use it as a tool to help you with your organism. So this will have to be done.
So what you're saying is that one there, we don't have to do the tests to build that out.
Oh, you look up the tests.
Okay.
You take every test that's pretty small, but you take every one of these tests and you look in the literature, and you find out for every one of these organisms, and that's a lot. So maybe your table, you say, you do these 5, and you do these, and you do these, and stuff, and then you come all together, and you've done it, and you fill in the table. And so that way it helps everybody, because nobody probably right now knows what you have. You might know, maybe it could be like this one, or this one, or this one, but you don't know. So I guess you could be with an attitude that says, I will only do my stuff and let the table worry about themselves. But you really want to get everybody into doing it, so you get it all finished. Okay, so anyway, that's that, but you do it however you want, whatever you guys decide, and you'll decide by table what to do. Okay, so let's look more at the project, and this is just the project work. So I think that's all they actually have on the project. The rest of it is things that we've, you already know, the tests that we've done. You've got day 1, day 2, day 3, day 4. So each of the tests, you'll just be doing day by day. And again, if you didn't do it on the first day, reading or listening to the videos before you come to lab is really, really helpful. What it does is it just gives you one leg up on you know, kind of knowing what you should be looking for and how to do it, and do you stab to the bottom or something like that. You're still reading the protocol as you're sitting in lab, but if you've already watched the videos and stuff like that, you have a better idea of what to expect. Okay? So that's really all the time I want to take for a lecture on this, because this really should be done in lab, but it's really hard once you get in lab, your whole focus is on your experiments that you want to do. It's hard just to stop and focus on this. But now that you've looked at it and you get a good chance to read it, and maybe you look at some of the reports, then you'll maybe have some ideas of what you have questions on. Okay, but that first one that we had where it talks about the actual report, it really has very detailed. I don't think I had hardly anyone ask me any questions. They just referred back to this document on the unknown report. And I guess at the very bottom I can show you. They give you a Berge's manual. That's something that we have in lab. But we also, it's a good source for looking up organisms. I think on the bottom of that same one, I keep opening it up. List of possibilities of your unknown. You already know what that is. And then the unknown report. This is the assignment link for that unknown report right here and here.
There's nothing there.
Okay, so we already saw what the report parameters are. So I don't know if they just take it off or what. And then way down here at the bottom, see they have some organisms listed, and they have scanned pages from the Berge's manual. And if you look at it when you click it, it's obviously old, but it doesn't matter because Berge's manual is already 50 years old. It's just organisms don't change. If you click on these, there could be 110 pages. So you just click on it and then you can scroll through it. Nobody needs to print it. You just scroll through it and you find your particular organism, and then you can learn information about the organism that you think you have. And that also may lead you away from your organism, saying, oh, okay, my organism didn't do that, or it wasn't this color, it didn't have this kind of precipitate or whatever. So these are very, very helpful. And I think here it says, here's a helpful site with a lot of test results for organisms, but they only test a few strains, so it's better to look at it in Berge's, and this is where Berge's is up here. But this is also another website that you can go to that has lots of information about organisms. But you really have to have a little idea of what organism you're looking for. You can't just read the whole thing. You want to have some idea of what you think out of this. We only had, I don't know if we had all 20 that we had for ours, but you only have 15 of those, of the ones that are available. So you might be chasing something that isn't one that we have, but you won't know that until the very end. Okay, so any questions about where you can find information? And I mean, it really, you think it's not very soon, but I mean, some of the stuff is due like May 13th or something. And I mean, we are getting towards the middle or end of April, and voila, it's going to be May, and then it's just going to hit you all at once. So be really diligent and try to get some of the little stuff done. And because of your presentation, you will, when you do your presentation, every figure that you put in your presentation will be your own. So you will take pictures of your organism, what your results of your organism is. You will not, when you're putting up your results, to say, oh, I had a positive test of this, and I had a negative test of this. These are how mine turned out, but the literature says it should be like this and like this. But this is what I got, and you have to put up your own. You can't go and put the literature and say, this is what I should have. You have to put in what you got yourself. So no matter how good or bad your results are, you want to put that up. The same thing with the gram stain. You'll start out with the gram stain. You will show your gram stain, not the gram stain that might be published for that organism, so you show yours. So that's why you want to take pictures of everything that you do. Okay? Makes sense? So any questions about this? I've already taken like 25 minutes, so.
So the presentation, is it supposed to be like a PowerPoint presentation?
Yeah, it'll be a PowerPoint.
Okay.
And I think, I don't know how many slides people had, like 10 slides or something. to do it. And we'll decide when we kind of see what all the unknowns are. 'Cause supposedly, you will all have a very specific unknown to talk about. So there's no duplications. But it kind of just depends on how it turns out. All right, so we're going to go off to here, and then we're going to go on to the lecture so that we don't have such a long evening. Okay, so okay to turn this off. And again, you have all of this, anytime in lab, you can just pull this back up again and we can look at it. I do have some of the things that they did from last semester. So if you kind of need some formatting or something, it's exactly the format that we have here. Some of them, the only ones that I've got to keep are not exactly perfect. Like this one does not have more information in here like it's supposed to have. And then this is the rubric for the unknown. This is the presentation rubric. So you'll get this, and this was worth, this was worth 50 points. Sorry, so this presentation rubric, presentation was worth 50 points. I think it's still worth 50 for this semester. I'm not positive. Okay. And the paper itself was pretty small. So here was their tree, decision tree, and then this was their 2/3 of the page and their references that they had. So it's not really onerous to do, but there's a lot of work that goes into it. So you make sure you start early. All right, so let me get out of this, and then as you start looking at things, we can talk about it a little later. All right, the talk that we're going to do today is on epidemiology. So this is chapter 14. Some of this is, let me put all this here before we get it lost. Some of this is directly from the chapter, some of it is stuff that I've put in there, so I think it will follow the chapter pretty well if you're using a textbook or something to follow it. But I think there's maybe 40 or 50 slides, or maybe a little bit more than that, and we will have to decide when you've had enough. We'll take a break probably right at 7 o'clock, and then kind of go from there to see what we need to cover, but I'd like to cover the entire chapter. So again, epidemiology, it's really based on when there's an outbreak of something. We get epidemiologists in, and what they're trying to do is they're trying to find the index case, what's caused this, how they can alleviate whatever the source is. So they try, if they can find the index case, then they can try to find the source. And once they do that, then they can figure out how they can alleviate it altogether. They can use some drugs or remove something like the pump handle, etc. So this kind of goes back to Snow. And Snow, when they had the cholera outbreak in London, and he went kind of street by street to see how many deaths were in different areas, and he kind of corralled it to a certain street that had more deaths than other ones. And then he started doing interviews. What he was looking for is the index case. Where is the first case that started all of this? And he realized it wasn't really a case, it was an area. And that area was because they were getting water from a certain pump. And the pump, as it was bringing the water up, now every pump in the whole city brought up water from the same source. So it was not the water itself, it was something that was being put into the water after the main water flowed through the city. And that's why he went to the pump, and he found the pump handle had something inside of it. As the water flowed through, it flowed past this area of the pathogen, and the pathogen got into the water, and of course the pathogen could multiply in that area. And so once that was taken home, put in a refrigerator or whatever, or in a bucket, it just multiplied and multiplied, and people who drank from that water then were getting sick. So it was really kind of a hard case because all of the water in the whole city was being used and not everybody in the whole city was getting sick. So we had to really narrow it down, and that was what really brought on epidemiology. So this sentence at the very beginning is really important. It's probably really important today as it was then. It says healthcare matters to all of us some of the time, and public health matters to all of us all of the time. And I think we're seeing that now in maybe the present government, where we are trying to narrow down what do we really need as a society, how much money should we spend for different things, what can we do without and still keep society safe, and that's one of the parameters. But that is really what's happening. We're eliminating some things that we are used to, like we could possibly eliminate the CDC, and we could eliminate other government agencies that are in charge of epidemiology. And so if that happens, we have to think, what is that going to do to the health of everyone? Maybe your own health you can take care of, but if you are really healthy and you go into an area that people are sick, the likelihood of you being able in your own way of keeping yourself safe is not probably going to work. So you're going to have vaccinations, you're going to have some kind of medication, you're going to have someone find whatever source of this disease is going around to be able to stop it. So we'll go over periods of time to see what safeguards the society actually needs, what safeguards will we keep, and what will be the outcome of that. So I think this chapter is really kind of prominent for today. So, okay, and this is just an old clicker question I put in there. Let's see if we can kind of go through this and refresh our memory about HIV. So we're looking for the thing that is false. And then maybe you can tell me why is it false. So these are in there. Okay, so anybody got any things now? Okay, so we've forgotten a little bit about HIV, I can see. So let's just start for the first one. Life cycle includes a provirus. Okay, provirus, what does that mean? If it's a prophage, what would it mean? The phage would integrate into the genome of the bacteria. So provirus means that the virus integrates into the genome of the host. And so that's what's so dangerous about HIV, is because it does. And so that's why HIV stays with a person for their life. It is not something you can cure. It's for your life. You will take drugs for the rest of your life. And so that's another really dangerous thing. If we pull funding from HIV, people that have HIV, it will then become very active, and anybody they're exposing is going to be active, and pretty soon HIV will be right back where it was. So that's really an important topic that we all have to be watching. All right, so this one, it is a provirus. So it actually goes into the genome. And I think we mentioned also what genome, every cell has its own genome. So what cell is this infecting and gets into the genome of that particular cell? T cells, yeah. And T cells, we will do this in the next lecture, or maybe the one after that. T cells are your main immune cell. And what it does is it regulates the rest of your immune cells. So probably everybody has heard of B cells that make antibodies. Well, the T cells always regulate the B cells to say whether they should or should not make an antibody against whatever they're displaying. So the T cells are really the regulator of your entire immune system. So if HIV infects that, then that's why it just decimates a person. And if you can take the drugs enough to keep the virus down, then your T cells are high and your immune system is strong and you are doing well. But if your immune system gets weakened because of HIV, then you are all kinds of opportunistic infections hit you. And that's where they get this thrush in the mouth, where it's got like yeast infections all in the mouth. HIV is really bad just by itself. Okay, so let's look at this one. It is a positive strand RNA virus. Is it? It definitely is. And it replicates in the host nucleus. It does, because it goes from this single-stranded RNA to DNA, and that's where it integrates into the host. So the last one, this really sounds like it's okay, because it says uses replicase to transcribe single-stranded RNA into double-stranded DNA. If they said it went from single-stranded RNA to negative strand of RNA, this would be good. Replicase would be doing that. Replicase means RNA-dependent RNA polymerase. But this is going to DNA, so this is the reverse transcriptase that should be in that spot. So it kind of sounds the same, but it's not. So kind of a refresher on HIV. We bring up HIV again in another chapter. All right, so that was this one. All right, so now we're back to snow that we've already talked about. So microbes, this is all about epidemiology, which is microbes, and they are the ones that cause disease. So whenever there's a new outbreak of some kind of disease, and they don't always know what it is, they have to search for the microbes. And it's kind of surprising that our adult body, we have 3.3 pounds of microbes on us. I mean, they are just everywhere, which is good. And we're going to find out why that is good in a few minutes. So there's 10 to 1 microbial cells to every host cell. So you have bacterial cells, you have 10 to 1 to every single one of your cells, which is really surprising. So that means just in the gut alone, you have 100 trillion microbial cells. So we live with them all the time, and they're basically beneficial to us. But there are times when they're not. And we call them our microbiota, and I think we've kind of talked about that before. And they reside in us and on us. And the idea of in and on, when they reside on us, means that they're all on the exterior, eyelashes, tongue, teeth, everywhere on the outside. But every time you open your mouth, you're like a big hole, goes through your mouth and your esophagus, around and around, and then out the anus. So you are one big hole. And that big hole, air comes in, food comes in, everything comes in, it also goes out. So all of that is still open to the outside. And that means that whole area is covered with all kinds of bacteria. And that would give you then the understanding that just because we swallow something, if it doesn't get into our cells, get outside of the epithelial cells into the body proper, then it really isn't going to hurt us. This is going to be expelled from the body. It's only when it gets past those cells into the sterile part of the body does it start causing disease. Okay, so some of these microbes that we have a couple slides that will talk about the same thing. They have a mutualistic kind of relationship. So the host and the pathogen, we both benefit. And I'll show you a slide showing exactly how we both benefit. And then one of the things, this colonizing barrier, if you have microbes that are good on you, you have them all over you, and there's a pathogen that you maybe put your hand down and then you touch your body, that pathogen is trying to stick onto your cells, and you're having so many other microbial cells on you, your microbiota, there's no room for that bacteria to stick on. So it really is going to be washed away. And I think we saw that in part of the thing we tried to show when we did the thumbprints and the fingerprints. If we washed our hand, we touched our shoes or something, and then we imprinted, and then we washed that off, and then we imprinted again. And when we imprinted the second time, we still had bacteria on our hands, but not nearly as much. So the bacteria we still had on our hands was our own microbiota. It does not wash off as easily. But the transient bacteria that hasn't got a chance to take a foothold on your skin yet is the thing that got washed off. So you saw a whole bunch, and then it was pretty much washed off, except for your microbiota. So that's kind of what we were showing in that exercise. So these pathogens, whenever they are on your body, you pick them up, they need to have nourishment. So they are trying to get to your cells, and if your own microbiota is excluding that, then these are not going to be able to stay. They'll just fall off and be eliminated. And we have what they call non-permissive microbiota. and they secrete like an antimicrobial substance. And when bacteria try to, pathogens, let's say, come in, not your normal microbiota, when it tries to get onto your cells, it's usually going to be removed by these antimicrobials that your own cells are secreting. Okay, and then we're going to talk about how we acquire our own microbiota in a second. So these non-permissive ones will make the environment of your skin unhospitable, and pathogens will leave. So, oops, we'll skip this one for now. So this is potential pathogens. Here are some potential pathogens up here. And these are epithelial cells that you have pretty much all on the lining of your body, so that when you want to go into the body proper, things have to come through here to do that. So here is where all the bacteria are, and this would be the inside of the body. So the bacteria have to go through, attach onto these cells. And you can see we have some, these over here, these are unfavorable for the colonization. These are favors the colonization. So you can see these will not let pathogens land on them or any of your cells. But if you have these as your microbiota, they will allow pathogens to accompany them and stay with the same area. So you want your cells, you want your normal microbiota to be non-permissive, so that it will not allow pathogens also to occupy the same space it does. And sometimes we kill our own microbiota, maybe with antibiotics or different things, and we'll talk about how we have to replenish that. So these pathogens, when they come in, they could be commensal. So the bacteria is going to benefit and the host is unaffected, so that's not so bad. They can also colonize and be parasitic, and then that's going to be bad. So the host will be harmed, but the organism isn't. So I have another slide coming up that will summarize these terms, so it will be a little bit easier to look at that. So some of the organisms that are excluded by the non-permissive are Lactobacillus and Staph epidermidis. So these are ones that we pretty much have in our body almost all over, and so they're the cells where we usually allow them to remain. And here is kind of a thing. Here's the terms that we just talked about. The relationship with the host, we have terms that we want to attach to that. So a lot of this chapter you're going to see are terms. So it is going to be a heavy matching our multiple choice chapter because it used to have so many different terms that we have to talk about. So indigenous, this has a permanent relationship. When we did the experiment and you touched your shoe and then you had a whole bunch of bacteria and then you washed that off with all kinds of substances and you touched the agar again, that was your indigenous microbiota. That is the stuff that you live with that permanently. And so it basically stays permanently on your body. And these are some of the ones that are there all the time. And then transient, these are the ones that when you touched it the first time, you had a ton of bacteria, you washed off your hand, and you touched it again, they were gone. And those were the transient ones. And that's the same kind of nomenclature that they used in the lab manual. And then these are the relationships that we have with the organisms. Mutualism, both of us benefit. Commensalism, only the microbe benefits, but we're not harmed by it. And parasitism, this is when the pathogen is going to damage the host. Okay? So the thing on here, these highlighted in yellow, I would like you to at least be able to identify, and that means not come up with a fill in the blank, but come up with a matching, where you can match these organisms with this title, these organisms with this title, and this organism with this title. Okay, so it won't be something that you have to fill in the blank, but it's something that you should recognize. So maybe just reading a slide over a few times would make that something that you could recognize. So Andrew, was I standing in front of you, and could you see the slide when I'm there? Okay. All of a sudden I thought, well, you're right behind me, so maybe you can't see it. Okay, so just so you can kind of see, I'm highlighting some things in yellow, and I'll try to tell you why I'm highlighting them. All right, so this slide is really busy and a little small to look at. But there's a couple of highlights I want to put on here. So mutualism benefits both, but commensalism benefits the host, or the organism, and the host is really ineffective. So we have a couple of things here where we actually get benefit from some organisms. If we look at the female reproductive tract, there's a lot of women get yeast infections. And so the microbiota that's in the vaginal area, if it is lactobacillus, which is usually pretty healthy, it will keep the pH kind of low. So if the pH is low, then yeast will not be able to grow and everything is fine. So this is something that probiotics are something some people take them to replenish their lactobacillus. So this is one you should know is beneficial. Here's another one that's beneficial. This is E. coli, and E. coli produces vitamin B12, which is something that the body needs, and also vitamin BK. It's an antioxidant. So, and not enough to supply us instead of vitamin pills or something, but it is one benefit that the organism supplies the body. So none of these do you have to memorize. This is just to show you that you have many organisms that are the same in almost every part of the body. So none of those do you have to memorize. Okay, this is just to kind of give you an idea that there's a lot of different bacteria in all different places. The only thing is here, lactobacillus here, and its benefit, and then E. coli here in the intestines for its benefit. And that's where E. coli is. It's an enteric bacteria. Yes?
Can some organisms that are beneficial in one part of the body be like pathogenic in another part of the body?
I would say no, but these organisms, the reason this organism is good is because it lowers the pH, but it could also cause disease. So it does have a beneficial factor, but it's not unheard of that it could also cause disease. So I would say that none of these are going to be to the point that they cannot be pathogenic. So anytime you get bacteria that is out of the realm of your outer body and it gets within sterile areas, which all of these are sterile areas, are going to be potentially pathogenic. And we see lots of horror stories about E. coli now. I mean, there are some E. coli strains that cause disease. They've mutated in ways that we have no antibiotics against them. So even something like E. coli. And that's one of the ones that we're really careful in. And you've noticed that your E. coli, what color does it grow? Kind of a reddish color? Is that what you said, or what?
Like whitish green.
Oh, okay. So yeah, it's a different color than the normal E. coli would grow. And they do that to show that, in fact, the strain that they're using in lab is not an infective one. All of that has been removed. But it could easily mutate, and when they plate it again, if it ever does not have that color that it's supposed to have, then they would know that they would have to replenish that. Yes?
So could the one that's staying in our intestines ever mutate, like just on its own?
The one that's in the intestines?
Yeah.
Every time a bacteria replicates, it is mutating. Every time. It's just that usually the mutations they're not beneficial. So you have 500 bacteria that have just replicated, and 350 have mutated. They're not very strong anymore. The rest of them are taking all the nutrients. They take over, and pretty soon these die off. So usually it doesn't matter. It's only when one actually has maybe something that's virulent, and then it starts taking over. And it has to be able to grow better than the other ones. Otherwise, even if it's more virulent, if the other ones grow better, it's going to take all the nutrients and that one will die off. But yeah, it's mutating all the time. It's just that usually it's, the genome is like this long, and this is only the only important part of the genome, and so it gets a mutation here and there and there and there and there and there. So it doesn't make any difference. Same like us, we get mutations in our genome all the time, but our genome, we have billions of nucleotides, and most of them aren't doing anything. So if we get mutations in those areas, it doesn't affect us. It's only hit and miss. Sometimes we do. Because look how long we live, and we don't get cancer, most of us. We live forever. Look how many times we've replicated in our body. So it's just, it does happen, but it's not a frequent event. Okay, so here, now we want to talk about what tissues are sterile. So these are the ones that are sterile. Okay, and the thing that I kind of talked about on here is, so I'm talking about a person here, and you open your mouth, and it comes down here, and then it goes around in here, and so then it comes out here. So this part all through here, this is all protected. Your body is around this area. So this is where the sterile area is, and this is where the pathogens are coming in and going out. So when we talk about the sterile area, so the bone, the blood, cerebral, spinal fluid, joint fluid, all your internal organisms that are all out here places, they're away from our own microbiota and any pathogen that is not already getting into a sterile area. So if it does get into the sterile area, that's when we have a problem. And so mother's womb, that's also all sterile. Indigenous bacteria are going to be introduced then, because in the baby, when it's born, it has been in a sterile area all the time it's been a fetus. So as soon as it's delivered, it is into a realm of bacteria everywhere, no matter how sterile the operating room is. So the first thing that you do, I don't know if you've ever been in where they have a birth, is they take the baby and they slap it on the mother. You know, they take her, she has a hospital gown on, they remove part of the hospital gown and they do skin to skin. And if she is too unable to be able to have that happen, they take the father. So they gown him up and stuff like that, and then they put the baby right on his skin. So they want the baby to get as much microbiota from that family as possible from moment one. And that way that bacteria will start colonizing on the baby and will protect it in the same environment that the baby's going to go home in. So maybe they have dogs, or they have, you know, whatever kind of air they have in the house, but all of that will start helping protect the baby because it has no immune system. So the mother will be giving it antibiotics through colostrum, but it's really a precarious time for the baby. So that's one of the things that they do to help start off the microbiota. Okay, so by 1 year, though, the baby's got its own, so it kind of depends if it goes to see the grandparents or it goes different places and stuff. It's going to pick up some microbiota kind of its own. Okay, so the sterile places, there's something it's important that you know, but I guess we can use this slide to see if you know that or not. Let's do this slide. So this is the indigenous microbiota. So you have to know the terminology for that. And it said found in all the places except. Okay, so we have a couple of different things. So the upper respiratory tract and blood is the two that we talked about. So blood is the one that's sterile. Remember, up in here, you take a breath, and what's going into your respiratory system is just everything. Okay, and it's not going to get into the lungs unless it goes past those cells that are protecting the airways before they go in. So these are going to be, microbiota is going to be found in all these places. They'll be in your respiratory tract. That's when you breathe in, you filter it out. The oral cavity, you open your mouth. The vaginal area, it's always open to the outside. Should not maybe always be, but, and then blood. Okay, so blood is the one that would be our exception. If it is, then we have blood diseases. Okay, so since we have so much bacteria on us, they can feel pretty protected by it, but things do go awry. It's not always helpful or healthy. And so there's two terms we want to talk about, infection and disease, and they are different terms. So an infection says you're going to have a microbe, it's going to come in and it's going to start multiplying and causing inflammation. And inflammation is when your body senses, your immune system senses that there is being an attack by some kind of a pathogen coming in. And what it does is it comes out of the bloodstream. These are macrophages, and it comes out of the bloodstream, actually goes through the walls of the blood vessels, and it goes into the area wherever the pathogen is. And macrophages are like Pac-Man. It comes in and just chops up and eats any kind of a pathogen that it finds. And then it chews it all up and it gets rid of it. Well, in that process of going out of the bloodstream into whatever area, like you got a paper cut on your finger, and that hurts like heck. And then you look at your finger in just a couple minutes and it's starting to get really red and it's getting swollen right there where you had a little paper cut. And that's because you had all the macrophages going right to the fingertip and it's trying to eat up all the bacteria that is coming in before it gets to the bloodstream. So when you first cut it and it bleeds a little bit, you still have a whole bunch of bacteria that is not in the bloodstream yet, so your macrophages are trying to come there and chew that all up so it won't get cause you a major infection. And so this inflammation, that redness, that warmth, the swelling, the pain is because as it swells, you have nerves everywhere throughout your body, and those nerves are being pressed on by this extra fluid that's in your finger or wherever it is that you've gotten hurt. So that is what inflammation is. It causes that red, warmth, swelling, and pain. And that's because the macrophages are coming there to try to help overwhelm whatever bacteria is coming in. So disease is when you kind of lose the battle. So just because you get an infection, you get bacteria into your finger or your blood or something, unless it can start multiplying and dividing, and unless your immune system cannot take care of it, then it can start causing disease. But your immune system is so fine-tuned and so good that most of the time, no matter what happens to you, it can take over as long as you can get enough sleep, as long as you can relax, as long as you can allow your immune system to take over all the energy of your body to be able to fight its battle, then it usually can win. But when we do 100 million things, even though we're really sick, can hardly walk, you're exhausted and something, you still come to school, you still do whatever you do, that's the time it eventually just gets overwhelmed, can't do its job. And then you get sick and then you have to stay home in bed, and then you get better again. Okay, so disease is one thing. Just because you get an infection, it does not mean you have disease. So you can get infected with something, but if your immune system can take care of it, you are not going to go to disease. Okay? Only when your pathogen gets a foothold. All right, so here are some kind of the same terms. So exogenous infection. So this would be from microbes that are outside your skin, let's say, your own microbiota. So it might be something I picked up off the table, I ate off a spoon that had some bacteria, something on it. So it would be that kind. So exogenous is something that is not your own microbiota that is breaching your defense system. And endogenous is when your own microbiota does. So a paper cut can be exactly like that. You get the paper cut and you have all your own microbiota on your finger and some of that gets in. But you also have other microbiota from maybe the paper that came in. That would be exogenous. So there's two different kinds. Exogenous means outside of your normal microbiota, and endogenous means it is your own microbiota that's getting into the sterile area. And opportunistic infections, these might be the commensal pathogens that you have just sitting around on some of your epithelial cells, and now they have an opportunity to cause infection. So they can take advantage of that environment. And some of the environment, the reason they can take advantage of it is your immune system, because your immune system is always surveilling your body to look for pathogens. There are structures and things on pathogens that you have something called toll-like receptors that are on all of your macrophages, and they go around and they are able to tell if this is a pathogen. So they look for standard things that are on pathogens. They're not like an antibody that is very specific to a pathogen. These are, there's only 10, I think, in the human body, and they only recognize certain things, like pili, maybe, because that's not on the human body, or maybe some parts of a flagella, or something else that is on a bacteria. So that is the way that we protect ourselves without really causing a big alarm system. If you have antibodies, they are made specifically to a very specific target. These are general targets, just a general bacteria. So if your immune system gets weakened, you don't have that defense. Let's say there's something happens to your microbiota, and that can happen because you take antibiotics, and we'll talk a little bit more about that on the next slide. And then if you have some kind of a piercing, and this is not a small little piercing, but this could be a knife. This could be something that really cuts a big hole in the body, and so you get a lot of opportunity for infection to get into that area. So that gives a lot of opportunity for organisms to then enter that and overwhelm your own microbiota and your own immune system. So let's look at this and we kind of go back through. This is some of those conditions that overwhelm these, or caused by these opportunistic infections. So again, we mentioned the weakened immune system, and then antibiotics. So to elaborate on that a little bit more, we take antibiotics for a lot of different things. So you go to a doctor, and depending how stringent your doctor is, he'll say, oh yeah, here's an antibiotic for that. And so you take that antibiotic, and sure enough, it probably works. So you feel better and everything is good. Maybe some of the advice could be just go home and go to bed. You know, for 1 week, you're just bedridden and you are going to get better on your own. You have a strong immune system and it will take care of this. But when you have antibiotics, it targets not only the pathogen, but it's going to target all your own microbiota too. Because as we see on bacteria, if you're gram-positive, you look like one thing under stain, and if you're gram-negative, you look like something different. That's exactly what those organisms look like to an antibiotic. The antibiotic doesn't know exactly what kind of gram-negative or what kind of gram-positive it's targeting. They have some that the antibiotics will target the ribosomes. Well, they don't care what ribosome it is. It's just a ribosome of a bacteria, which means your own microbiota can also be included in that. And if that happens, then you are getting rid of some of the microbiota that might be a non-permissive organism that's on your body, and now you're having other pathogens come in. So antibiotic use, it can also cause the ability for opportunistic infections to occur. And then HIV patients, they get this thrush. I had mentioned that, and this is what it really looks like. So it's not at all pleasant. It's just that their immune system is so weakened that opportunistic infection with the Candida is one of the ones, and that is a yeast. Okay, and so I would want you to know, be able to match this name with thrush, and that it's with HIV patients. Okay, so just kind of a matching thing. I would not ask you to fill in a blank with that words. Maybe I could give you these two and ask you to fill in HIV as a blank, but not either of these two words. Okay? All right. So infections in the blood, so it's one of the things that if you get an infection in the blood, that is a non-sterile area. And so, or it is a sterile area, sorry, and so getting an infection in that is usually a cause for disease. So infection in the blood is anemia. So if it is a bacteria that's causing the infection in the blood, it's going to be bacteremia. And if it's just bacteria that is overgrown and really taking over, it's a little bit more serious, so it's called septicemia. And then viremia, if it's a virus that's causing this, and toxemia, if it is some kind of toxin that's in the blood. Okay, so we have several different emias. And this is something you don't really have to memorize. It's just to show you almost all organisms are going to be involved in this. Okay, so emias are just where we have living organisms in the blood, and it depends whether it's a bacteria or whether it's a virus or if it's a toxin. Okay, so I think we might be at a good stopping spot. So maybe let's stop here. It's only slide number 15, but it would give us maybe like a 10-minute break, and then we'll talk maybe, and it might take us till 8 o'clock, so that's kind of an estimate. So we'll take maybe just a 10-minute break here and then come back and finish as much as we can. Okay? And then if at this time you want to maybe look at the exam, if you have not looked at that, so that's here. And if you have any questions about maybe what we talked earlier about, I have some samples from the previous, air quality pill, some previous ones from last semester. Oh, these are cool.
I know you talked about, and this is off topic,
Uh-huh.
you were discussing a little bit, white blood cells.
Mm-hmm.
So neutrophils are white blood cells.
Yep.
So what is their job? Because I know you talked about for the others.
Yeah, neutrophils are first responders. So what they do is they come to whenever there's, let's say there's a cut and there's signals that are being sent through. So macrophages usually are going around in the bloodstream all the time, and the macrophage gets the signal, and so then they signal to other molecules. Oh, perfect, I'm with you. I just want to put this away, otherwise I will have it in my hand and not put it in the right spot. So neutrophils are called to the spot, and neutrophils only live a couple of days. So they are willing to risk their whole life to capture this thing. So if there's, let's say this is a bacteria, so it will come and it will surround the bacteria, and then it will just explode and kind of kill itself. And it will spread out all over your desk, so it will get this.
Hi, Vince.
It will get everything it can, and it will be like a net. It's called a neutrophil extracellular net. And that allows it to then capture as many bacteria as possible in this net. And the macrophages come over and they go.
I think I saw it on the camera.
So the neutrophil, that's their first job, is the first line of defense.
It's interesting because my cat is not well right now,
Oh.
and he has his lungs done, and the neutrophils are like falling apart.
Oh,
Yeah.
so they're really trying to do their job. Yeah, I mean, it sounds like it, because your body will replace more and more of them. So it sounds like your the cat's body is trying to fight disease. Yeah, whatever it is.
He's given broad spectrum antibiotics,
I think it was on
but it's not really.
It's the rest.
I saw some crap that said that in
Yeah.
there too.
It is better finding birdies as possible.
No matter all.
Okay, that's where I saw the word. I have no idea. I was like, I swear I saw that word.
Yeah, I checked it on my offer up, I couldn't find
I
it.
mean, that's the way, I guess,
What
the
do you think is
last
it?
few, I'll get rid of
It's
that.
just help set up this page.
Like a logic tree type
Know exactly what.
thing for bacteria.
Yeah.
Just to help
It would
with,
be hard to tell because,
Just to help with
for
one of
some
these
reason, the body is.
like it'll say like it grows on this, you know how we had
But
that
what is it
no
fighting
project?
against?
So it's like a test,
Yeah.
test checklist for various.
Yeah.
No, and they're really, when we
Pretty
get to
much.
that, when we talk about it's so interesting, and it's really something to able to realize how.
I mean, honestly, you can almost
fit
take a
your
picture of
body
it and
is
say,
to defend
fill in
itself.
the blanks with all the
Yes,
types of
and the
tests.
only thing that we have to watch out for is we eat the wrong
And then
thing,
double check
we
it.
don't
I'll
sit
double
around,
check it.
we Don't give it to
That's what I'm
anybody
hoping.
to do its job.
Because you only need to do that, because that's basically just our reference
I
sheet.
get the same emails I do about wellness
But then,
days.
like, also, I know one thing I was like, I started crossing
So
them
they
off.
encourage students to recognize when they're not feeling well or whatever, and then take time off, take time to get better. And to have maybe professors understand if a student says they're sick, they're not coming, you know,
I
don't
don't
give
think your
them
tools
punishment.
sit with us.
Say, this is what needs to
I was just gonna do the whole thing. I was just gonna do ChatGPT and then check them all.
Oh, yeah, that's a good idea. Yeah, then they,
Because
but, you know,
I'm
it all
fascinated.
goes on this honesty.
Yeah.
This is really how you feel, and be able to,
So you got shit to do right now?
do what they have to do to take care of themselves. But it also maybe, they miss too much class, and so they
Come on, I
drop the
thought
class.
it
It's part of.
I was thinking, I have like Slither.io here.
I was trying to see what you were watching.
Oh, I was watching, I was watching 90 but I got bored.
You're watching what?
Dragons would not slow down and rest.
It's like a spin-off. Did you ever watch Grey's Anatomy?
Yeah, it does.
My
That's why
girlfriend
this,
did.
when we, this
Fine,
class
yeah,
was
I had
from
to.
7 to 10 at night.
So there's
And it was horrible. And the students, you could
Oh,
just see
Grey's
that they
Anatomy or-
were.
911.
Oh, yeah, I haven't finished it.
Exactly.
Okay.
Yeah, I mean, even
It's a
if
good
it's
one.
3 nights in a row, no matter how far away or
I
how
go
close
and
you
watch it
were,
like 3
it's
times
still
on my house.
tight.
Does your girlfriend want to work in medical field?
She's an ER nurse right now.
Next thing you know,
You should
it's
tell her to watch the
11 or
pit.
so. and then
Yeah,
you
she
have
wants to.
work
She's like,
early
can we
and
start the
stuff.
pit?
Yeah,
I'm
you
like,
should.
I heard it's good.
no, it's because
Maybe one night.
Grey's Anatomy pisses me off
It's
because
not.
it's so long all the
When they
time.
want to go back
And I'm
to
like,
7 to 10,
and she,
The
which
pit
is crazy because she's in the medical.
I
She
can do
doesn't
it.
care. I'm like, that's wrong.
Okay, yeah. Grey's Anatomy is like so inaccurate, but the pit
They
is supposed
say
to
it's
be really accurate.
closer.
Yeah.
I would still nitpick. I'm certified, like, it has to be accurate, hater, dude. That shit pisses me off.
Yeah. What hospital does your girlfriend work?
Shores in Long Beach.
Oh. I was gonna say, because I volunteered in the ER at St. Joe's, but not the same. Is it veterans?
And so is anyone looking at these slides on your computers? Because are these slides, anybody looking at them on your computers? Okay, and so they're PDF and you can write on it or something? Okay, so I tried to make it on something that you could write on. I couldn't.
Yeah, the PDF is.
Okay. Yeah.
Oh, you still looking?
Wait, you do?
What are you looking for?
$10? No, I'm not going off.
For what?
Burglaries or something?
So on this one, it says you should reference
The burger thing?
Yeah.
Oh, yeah, it's on
this one.
the first link.
This one?
I click on it. Click on it.
But it's only for this one.
No, scroll down. It has like a few of
Does it
them.
have like an embedded link?
Here, but like, scroll down more. So for like, wait, go back up. So for that one, it'll tell you, like, this one's on our list, so it'll tell you like all the stuff you need to know.
Okay, so all of this would be for our stuff.
I think so. They should all be in there.
So those 3 links or whatever, or 4 links are the Burglaries
Yeah.
sections that cover all your stuff.
Yeah, so that's where we can like double check everything.
I was over here like looking for this on OfferUp and all that for the PDF.
Shit, I'm glad you didn't buy it.
Oh, I wasn't gonna. I'm like, no, if it's 20, I'm cool.
Dude, if it's freaking any money, I'm gonna find a freaking pirated PDF.
I don't mind.
You were quick enough. I was just like, alright, I'll take Vincent.
I don't mind paying 20.
Did you buy the textbook? Like, is that how we got the PDF?
Yeah.
Oh my gosh, do you want me to bet?
He's the goat, dude. I know, everyone in the class owes him like
I was
$2.
like, shit.
Dude. Well, thanks for doing that. That's nice.
And I'm saying I have like the anatomy book and a physio book if you guys need it. I don't mind sharing these.
How's anatomy for you guys?
I'm done with it.
Oh,
It's cool.
yep.
We just did the sheep brain dissection.
Oh, you did? Oh, that's a fun one.
So are we kind of ready
I
to
was so hungry.
start again?
Just kidding. I'm just kidding.
We need a little more time.
I'm so good.
Break for your mental health.
Well,
One more.
I decided to go back to school.
Okay, so let's go ahead and get going again. So we're gonna carry on, and again, here's some more back here. So we got through the EMEAs. So now some more terminology. So infection, and then going to disease. Remember, just because you have a pathogen that enters into the body and it even starts to multiply and divide, as long as your macrophages and your immune system is taking care of it, you really don't have disease. So only when your immune system cannot handle it, and then that's when we actually go to disease. So we have ways that we talk about these types of infections, and the infection doesn't necessarily mean disease. But primary infection, this is really when you're healthy and you get some kind of a pathogen, and it starts multiplying in your body and causing disease. then this would be considered a primary infection. So this is the thing that is actually making you sick. A secondary infection is when the body already has maybe is weakened by something else, and HIV is a lot of things we bring up. And so when you have AIDS, then that's anything else that you get, maybe this thrush or something, that is considered a secondary infection. And then local diseases, this is like, it's just maybe a single area that the disease is in. And a lot of this is you think of maybe you get a boil on your finger, and that spreads to the finger, and this is where you get all infection and stuff, but it doesn't really spread to the rest of the body as long as it can be taken care of. So that could be like a localized disease. And the systemic is when it gets to the bloodstream, and that's usually the most dangerous kind because it's very, very hard to treat. So it gets to everywhere in the body. So this, a boil, if you don't treat it, it could be a local disease, and then all of a sudden it gets into the bloodstream, and then it's really everywhere. Yes?
So under primary infections, and pneumonia is not really, that's not really a primary infection, right? Under COVID-19, because people were dying from pneumonia.
Right. Well, they were really dying from the, well, you're right. It kind of depends on what stage they were, because what happened for the pneumonia is that they had COVID, and COVID was in the lungs, and so your immune system, like it should be, was going crazy, coming out of the blood stream, and then bringing with it a little bit of fluid every time a macrophage came out. So your lungs were getting more and more fluid, and then they got the pneumonia, and so really the cause was COVID, but the death was really related to the pneumonia. So your body, and as most people who got COVID, they recovered from it. I mean, and some of the people got it and they said, oh, I didn't feel too good, but I don't think I've ever had COVID, and they'll test, and they said, yeah, you have antibodies for COVID. So you had COVID in your life and you didn't know it. So some people didn't have too much effect from COVID, and other people did. But really, this, when you got into pneumonia, just like they did with the flu, the pneumonia was a secondary. It wasn't, I mean, it was a cause of death, but it was because the system was weakened already. Yeah, pretty sad. Yeah, and I mean, when you compare our results to what they had they always talk about the worst thing that's ever happened, the big pandemic, 20 to 50 million people, and you think here for COVID and stuff like that, we had an awful lot also dying, but our population was so much larger that you really didn't notice it. I mean, it was a very scary event, but hopefully we are going to be prepared when it happens again, because we know it will happen. Okay, so now we're kind of changing topics to say, how does this disease start? Disease is not going to happen in the body unless the target for the disease, which is some kind of a cell, the pathogen can target that cell, hang on to the cell, and enter the cell. And so the only way they can do that is they usually have to attach in some way. Just like when we looked at the viruses, we saw that the virus would attach to its bacteria, let's say, and when it attached to the bacteria, then it would be able to be taken in by that bacteria and entered. This attachment for the virus, the words that we had for that, we just called them attachment proteins, but in bacteria we called them adhesions. So the way bacteria will adhere is through adhesion molecules. And there's a couple other ways they do also adhere, but adhesions are kind of the overall term. And on the surface, they have lipoproteins, and so that would be lipids that are on a protein, and the same thing with glyco. So this is sugars that are on proteins are lipids, lipo and glycoproteins. And these you can kind of see down here. So here's a glycoprotein. This is on the cell itself. And then here is the microbe that's coming in, and it's trying to bind this glycoreceptor that's on the bacteria itself. So the organism itself. So this bacteria is trying to enter into that organism, and so it has to attach to something and then going to be brought in. And another thing, bacteria and viruses when they have receptors that they bind, these aren't receptors that were made on the cell for them. They're receptors that are made for other things, and bacteria have just multiplied and divided enough time that they have made a molecule on their surface that matches the molecule that is used for something else on the human cell. And we'll just call it human cells. So it wasn't made for an attachment by the bacteria or a virus. It is just now that either of those have mutated in order to use that same receptor. Like the spike protein on COVID, so it uses a receptor for the angiotensin that was already on the cell. Okay, so the ligand for the host receptor is always going to be very specific to the host, and this is what we call this, is a ligand. These adhesions are going to be ligands that are going to attach to the host, and it's very specific to each host. Bacteria can just attach to anything if they get in the body, and they're supposed to be attaching to your lungs, and you swallow them, they're not going to get to your lungs, so you're fine. If you breathe them in, they can get to your lungs, and so you may not be fine. But different bacteria have different locations that they are going to be able to attach to it. Same thing with viruses. So the only way we can get rid of them then, the two logical things is we could either block the receptors that they're binding to, and that would stop them from being able to bind and get in. But like I said, the receptors that we have in our cells that the virus or the bacteria bind to get into the cell is not made for the bacteria or the virus. It's made for something that we need to do every day in our life. So if you bind that or you block that receptor, you could be blocking something very important in the body that the body's doing and needs that receptor for. So that doesn't work. So what you have to do is you have to get molecules that will block the pathogen when it has something on its surface. This molecule, whatever it is, we need to get a molecule that's going to block this one. So when it comes in to meet its glycoprotein to get into the cell, this is already blocked and it can't do that. So that's always what they're using now, is they're trying to target something, but they first have to discover what it is that's on the surface of the microbe that is actually binding and where is it binding. So once they can do that, then they can fashion some kind of molecule to block this. And then the pathogen won't be able to enter into cells, and therefore it's not going to be able to cause disease. And causing disease is called virulence. And so if we can block the pathogen from using something to cause disease, its virulence, then it's considered avirulent, meaning will not be able to cause disease. Okay, so these virulence factors, and there are several things that a bacteria can have that is considered a virulence factor, because a bacteria itself is just proteins and lipids and things. So it's nothing that is dangerous, except it gets into certain cells and then it multiplies and divides in those cells, and then it kills the cells. So we have to keep it from doing that. The factors that make it so dangerous is it has to be able to attach the cell to get into the cell. And so these adhesions are the things that help it attach to the cell, and these are what we then call virulence factors. So these are things that a structure, molecule, or protein that aids the bacteria in getting into and causing disease in the host. So whatever that organism has on it that allows it to cause disease, get into the host, is something called a virulence factor. And there's a whole big list. There's 20 or 50 of them. But adhesions are one of them. And you can see why this would be good. Here are the adhesion proteins. They can help bind to the target cell. A whole bunch of them bind to the target cell. They start forming this biofilm on the outside. And you can see this cell now is being affected by this. That last cell is maybe the macrophages are going to that site trying to kill all these organisms, but it has an inflammation now. So one of the things, virulence factors, and there's many of them, one of them is adhesion proteins. Another one is, Adhesion proteins, they have biofilms. Those are virulence factors. Extracellular enzymes that are released by the cell can be things that will attack the pathogen or attack the human cell. Toxins can be released by an organism like cholera, releases toxins, and it gets into a cell and causes all kinds of diarrhea and stuff. Just all the fluids of the host will be eliminated and the host will die. And anti-phagocytic factors they have, like they can have the bacteria can have capsules or something around them, whereas the host is trying to capture them and bring them in. They're sticking to whatever it is that is bringing them in, so that allows them to get into the cell. So all of these kind of things can be what we call virulence factors, and that's that allows them to be more pathogenic. So pathogenicity, this is just how disease-causing a pathogen will be, and virulence is just the degree of that ability to cause disease. So a couple of different terms. So virulence is just causing disease, and the pathogenicity is the ability for that organism to cause disease. Okay, and then these are just kind of a picture of a whole bunch of different things. Virulent surface proteins they have. This is like a capsid on the outside. Here's some adhesion. Here's some enzymes that it secretes. Oh, and here's something that we'll talk about a little bit later is lipopolysaccharides. And which of our organisms, gram-positive or gram-negative, has lipopolysaccharides on the outside? Both of them? Is that what you said? No, it's only one. And it makes sense that's only one, because lipopolysaccharides have to be attached, as the name sounds, lipopolysaccharide. Polysaccharides means many sugars. Lipo means it's got to be into a membrane. And so probably gram-negative, because the gram-negative is the only thing that has an outer membrane. The inner membrane for the gram-positive, then is going to be stacked with all that peptidoglycan on the outside. So the lipopolysaccharides are not going to be showing. But on a gram-negative, the lipopolysaccharides are on the outer membrane, and they stick up from the outer membrane. And they cause a lot of disease, and so we'll get to them, I think, in a little while. All right, so this is just two terms up here that are really important, and these are just some of the things that are considered virulence factors. And these are just things that allow that bacteria to cause disease. All right, so any bacteria, and I think this is kind of a question before, if you have E. coli in your intestines, and we've had it there, half your life, can it ever become virulent? And this is exactly yes. So any microorganism, whether it's our microbiota or not, can become virulent. They change genetically. They mutate. Things happen. They pick up. A particular organism can pick up through the conjugation pili. It can pick up a maybe antibiotic resistance gene, or it can pick up other genes that will make it more virulent. This is one of the things they always say. They want to eliminate your hospital stay so that you're not in contact any longer than you absolutely have to be with pathogens that are typically in a hospital. So the shorter the time period that you spend in a hospital unless you absolutely have to, the less ability for any kind of your own bacteria. Let's say here's your own bacteria just being a neighbor, and here comes another bacteria that has a pathogenic virulence factor on its plasmid is being transferred to the bacteria that you have. Okay, so you want to avoid that. So virulence genes can be transferred to another microbe through a conjugation pili, and this is a pili that's come through here. And so you have to think of what are the chances of this ever happening. People say, oh my gosh, that could be really dangerous. And it is, but when you really look at it, how often could it possibly happen? Because this has to be in an aqueous environment. They have to get close enough together. They have to have this conjugation pili get here. And then it has a plasmid right here that unravels into be a linear piece of DNA. And it has to travel through here before that these guys get too far apart and this breaks apart. So the chances are 1 in a zillion, million, billion, trillion, quadrillion. But you think one bacteria in 24 hours makes about 5 billion bacteria. So it doesn't take long to have a whole bunch of bacteria made. So even though the chances are very small, it is happening all the time. It's just really kind of amazing that that more transfer doesn't actually happen, and we have more antibiotic resistance genes out there. So anyway, any microorganism can become virulent just because of the ability to pick up DNA from other organisms. All right, so virulence factors, we're still talking about them. So collagenase, this is an enzyme that breaks down collagen. So you can see, here's the bacteria. This is on the outside of your epithelial cells. Here's where it wants to get in. So it can actually secrete a substance, and that will break through these this little area right here. So this is hyaluronidase, but it actually does the same thing. It breaks through this little area here, and then it can get into the main part of the cells. So this collagen layer that we have right here, that usually inhibits it. So when the bacterium secretes a substance to get through here, it's also going to have to secrete this collagenase. And that is an enzyme that's going to chop through this area right here to be able to get into the cell itself. Okay, and then this is hyaluronidase. This is another enzyme that is secreting this A's on the end of it. It means it's an enzyme. Here it is, A's. So it means it's also secreting, and what it's doing is it's cutting right through here, and it's going to get through the collagen layer because of that collagenase. Okay, and that's just once the bacteria gets in, it can just get into deeper and deeper into tissues. But not if you have a great immune system, because your macrophages will be right there to intercept it and chew them all up. All right, so here's another one. So coagulase is something that we've talked about, and we've actually seen that in the lab. Bacteria will produce coagulase, and it makes a clot-looking structure. And this is done usually when a bacteria wants to enter into a cell. So here's the bacteria, it's going to enter in the cell. There's like a little cut or something. And so if the bacteria enter now, you can see, here's the red blood cells, and then we will have macrophages right here within nanoseconds. So the bacteria know that if it gets into the cell right then, they're going to be chewed up, because all of the alert signals have been sent to the body saying, send the macrophages to this particular place, we have a paper cut. So what will happen is this coagulase will be secreted, and it will make a clot. And so that clot will help seal up this area, and no other pathogens can get in, but the pathogen that wants to get in is now inside this clot. And then when it's time, they secrete this kinase, and that kinase will chop up the clot material, and then on the secret, it will be able to go right in that same place where the clot formed to keep anything from coming out. And now the macrophages are gone. And now as soon as the first few of them get in, the macrophages will recognize again. We now have a pathogen among us, but they at least have an opportunity to be able to infect. Okay, so a couple of different enzymes that are really important as virulence factors, and then are part of the idea of virulence factors. So here are some toxins. So we have exotoxins, cytotoxin, neurotoxin, terotoxins. So these probably make sense. Exotoxins, this is secreting outside into the cytosol. This is neurotoxin, and it can be causing neurological problems, and this is like into the gut. So those little prefixes will tell you a little bit about what the toxin does. So we have exotoxins, means they're secreted outside the bacteria, and we have endotoxins, and here is our lipopolysaccharides up here. So these lipopolysaccharides are all on the outside of a gram-negative, and they signal to the macrophages that we are being attacked by a bacteria. So this is one of the things, I mentioned something called a toll-like receptor. The toll-like receptor is on the macrophage, and the macrophage, when it comes here, it feels these, it kind of, with its pseudopodia, reaches out and it feels these, and it says, okay, this is a bacteria. It has lipopolysaccharides on it. So that would tell the body, you have one bacteria that I have found, and I'm going to take care of it. Then what happens is you take antibiotics. And when you take an antibiotic and it kills a gram-negative bacteria, it kills this bacteria, the bacteria starts to fall apart. And when it falls apart, all these lipopolysaccharides drift off, all into your bloodstream. So then your macrophage comes over, and the macrophage finds another one. It goes, oh my gosh, here's a bacteria. And it finds another one. It goes, here's another bacteria. And it finds another one. It has another bacteria. And so it starts going crazy. It sends out signals over and over again to have more and more macrophages come to that location because they think billions of bacteria are now invading, when it really was one bacteria that fell apart and all the lipopolysaccharides floated away. And so each lipopolysaccharide is alerting a macrophage that there is a disaster at that area. And what happens? You go into septic shock. Because every time the macrophages come out of the bloodstream, a little bit more fluid is leaving the bloodstream. The bloodstream is getting thicker and thicker. It's harder for your heart to pump. Next thing you know, you have septic shock. So this is one of the big things they had, maybe 15 years ago. They discovered, and now they really are careful with antibiotics, because that definitely could happen. But anytime you kill gram-negative bacteria. Okay, and so these are just the examples of some exotoxins and endotoxins, but this is one of the major ones that have caused lots of problems. And there's a couple things. These are capsules. We know that here is the pseudopodia that I was talking about. Here's a macrophage, and the macrophage is reaching out like this. It's taking some of its membrane and reaching out like this, so we call it a pseudopodia, meaning a pseudo foot. And it's reaching out, and inside here are toll-like receptors, so they're little receptors, so it's feeling this. It says, what is this? Is this something I should take in and kill? Should I send signals out to the rest of the macrophages? But this one, it goes, I don't know, I don't know what that is, and it spits it out. So having that capsule around with sugars on it makes the macrophage say, I see sugars all the time, it's usually not pathogenic, I don't know what this is, and so it just spits it out. So this is kind of protective. So it's like an anti-bacteria defense. And then here is also they can go through incomplete phagocytosis, so they come in, but they don't go into this area here where these lysosomes will have acidic material that would kill the bacteria. So somehow it stops this breaching together where they would come together as one. And so it never gets this acidic material dumped into it. And so now the bacteria comes into that vacuole and then can multiply and divide and stay inside the cell without being detected. Okay, so here's a macrophage that took it in, hoping that it would be able to destroy it, it would be able to send it from here to here and kill it. But there was something that the bacteria had that inhibited this portion going in and joining, and so now it had this whole little vacuole for it to multiply and divide in. So bacteria have lots of ways to inhibit the effects of the cell to destroy it. And here's a couple of words. This fusion of the lysosome and with the phagocytic vesicle, that's what's happening right here, where it won't fuse together. So that's a defense. And the other one is these leukocidins. These destroy white blood cells. So these can be secreted by some bacteria, and that will actually destroy white blood cells. So there's lots of things that bacteria have to defend themselves, but there's a big arsenal that our body also has. And then toxicity, these, I mentioned cytotoxins, so on the previous slide we just said cyto, but these are cytotoxins, these are neurotoxins, and these are enterotoxins. I really only want you to know the name and not a whole bunch about them, but cytotoxins, in the cytotoxin means these toxins are secreted inside the cell in the cytoplasm, And these are where leukocytins are, and leuko is white, so these are white blood cells that it's going to be killing. And hemolysins, these are when they destroy the red blood cells. And we've already seen those kind of enzymes when we have the blood agar plates. So these two, again, kind of a matching thing. Cytotoxins, just this leukocytins, and those are white blood cells, and hemolysins, those are the red blood cells. And then the neurotoxins, this is going to hit the nervous system, and botulism and tetani. So these are things that cause rigidity, but you don't have to know the names. And here's enterotoxins, and these are ones that affect the gut. So entero, you should know it's the gut. Neuro is the nervous system, and then cyto would be the cytosol. Okay, and then of course we have antitoxins. Whenever we see one of these toxins, the body will be able to produce antitoxins. Many of those are just man-made that were given. You think of a snake bite, we have antitoxins for alleviating snake bites, or alleviating the molecule that the snake secretes that is going to harm the human. We just have to get it fast enough. And then these are toxoids, so these are ones that are going to be like toxins that are destroyed within the lab, and then we give a person a destroyed toxoid, or it's really a toxin that's been destroyed, and now it's called a toxoid, and then your body will make a defense against that toxin because it sees it, it thinks it's a pathogen, and so it makes antibodies against it. So if you can do that, then whatever that toxin is that you get, you can be spared from whatever the effect it might have. So these are called toxoids. And so endo, like the name sounds, this is a lipopolysaccharide that I talked about before. Sometimes it's called lipid A, but it's a lipopolysaccharide. This is an endotoxin, and this is, I already mentioned that it causes toxic shock. So this is one that you should know, a lipopolysaccharide. You should know that it's an endotoxin, that it's on gram-negative bacteria. Taking antibiotics that are against gram-negative can cause toxic shock because the lipopolysaccharides that are on the exterior of the bacteria will kind of all fall apart. So let me just go right back there again so you can see that figure. So here they are. So when this membrane falls apart, you're going to have all of these, and this is just a small portion of the membrane. There's just thousands and thousands of on them. So then it's going to make it seem like you have thousands and thousands of bacteria, and your body will have elicit a response, and the response will be overkill. It will be like we saw with the COVID, or we saw with the flu in the 1917-18. So your body can also go berserk, and when it does, it usually causes harm. All right, so that's what this whole endotoxin part is. So that particular slide is pretty important. Yes?
Would like allergies to things be considered any types of like toxins?
We don't really cover it in this course, but it is a response that your body has to something that is harmless usually, but for some reason there has been something to trigger a response, and every time that allergen gets in your body, it's going to have that same response. And so they usually do it like you do a little puppy. When that puppy cries because you leave, you go and you leave and you come back. And then you leave a little further and you come back. And you do that for maybe a month. And pretty soon the little puppy said, oh, they're coming back all the time. I don't have to make a fuss. So they do the same thing. They try to give people a tiny bit of the toxin and then and then a little bit more and more. So they're trying to build up the immune system so that it won't recognize this toxin as something bad, or the allergen. But what a pain. People who have allergies, it's really a bad thing. So it's just so uncomfortable and so sometimes unpredictable, especially if there's air allergens or something. Yeah. All right, and then this is just kind of a summary. These are some of the enzymes that we've talked about, and these are the sources of them. This is what they do, and this is the effect. This, again, is more of a multiple choice, kind of, bringing several things together. Okay. And these slides are all posted, so you can find any of these at your leisure. So, I mean, some of these we know what they do. Here's our kinase we talked about this going through, and leukocidins and hemolysin. So it seems like a lot, but we've talked about each of those. In fact, even dealt with some of them in lab.
Do we have to know the source, or just?
No, not specifically a source. Well, I'm sorry, the only source I might connect two together is coagulase, because we did experiments in lab with that. So that would only be the ones. But these kinase, we didn't do an experiment. Hemolysins, we did experiments with staff on it, but it would just be that it lyses red blood cells, so I wouldn't have them. So the only one I would expect that you would be able to connect the dots would be this one. Okay, does that make sense? Okay. And then this last slide, this is just a whole bunch of other kinds of toxins, nothing that we're going to talk about in any more depth. But things like we've talked about anthrax before, and these are just a whole bunch of different toxins that are out there. All right, so this, there's only a few more things I really want to cover, and this is one of them. This is extremely interesting. So we have a killer immune system, and so pathogens know that. And they need to avoid being into the cytosol or any area where your macrophages would be, because your macrophages are circulating through your body every moment. From the moment you're born until the moment you die, you have macrophages going through trying to do their job, and many other molecules that we'll talk about. So pathogens know that if they are remaining in the cytosol, or the place where your macrophages are, that they will be attacked and they will be killed. So when pathogens get in, they want to get right into a cell. As fast as they get into your body, they want to get inside a cell. Because macrophages don't go inside the cell, they just circulate around the cells. So here's a very interesting case. So this is a particular pathogen, and here is the macrophage. You can see the macrophage is recognizing it right away. Here is the pseudopodia that come out. There's toll-like receptors in here, and that toll-like receptor is recognizing this pathogen, and it's going to take it up and put it in this area and chew it up. So the Filopodia, that's this thing right out here, the little foot that comes out. It has toll-like receptors on the surface. It's recognizing the bacteria by something that's on the bacteria, and it's going to engulf it, and it's going to put it into a phagosome right in here, and then what it's going to do, that's going to merge with that lysosome that has all the acidic material, and it's going to kill the pathogen. So pathogens know that this is what's awaiting for them, so they need somehow to evade being destroyed. So this is a good slide to understand. We've talked about a lot of them. We talked about Filopodia, toll-like receptors, macrophages, phagocytosis. We've talked about all of those individual words. So this is a good slide. And this is one of the things that pathogens have done to be able to avoid being taken up. So here is a big cell. It's hard to see, but there's a big cell right here, and there's a big cell over there. And here comes a pathogen. This happens to be Listeria. And Listeria is, what it does is it zips right here, Listeria monocytogenes. This is like in ice cream and stuff. So Listeria comes right here and it zips. You can hardly see it, but it binds to some receptors on the cell. And when it does, it's stuck onto the cell now. So the cell recognizes, oh, there's something that I'm going to bring in, and it's going to put it into it goes through phagocytosis. It brings it in, and it's supposed to put it into this phagosome. So here it's got its membrane circled around it. It's in a little vacuole now. And before it can take this little vacuole and put it with the phagolysosome so that it can be killed and chopped up, the bacteria, Listeria, actually secretes a substance that breaks down the membrane that it's housed in. So now the Listeria is free inside the cell. And so it starts multiplying inside the cell. It's hidden from any kind of macrophage, and the cell doesn't know what's going on. So it's just having this bacteria multiply and divide. Then the bacteria says, Listeria says, I'm not going to be able to stay in here forever, so I need to move and get to another cell to infect another cell. So it encourages, it sends out a signal to the bacteria to make an actin tail. And this actin tail is just polymerized by different materials, and it makes a kind of a hard substance that will attach to the back of the bacteria. And this is all in small segments, and it can fall apart as it can come together. It can polymerize and then fall apart. So the longer the tail gets, the more momentum it has for this bacteria. It's pushing, as this tail is being made, it's pushing the bacteria against the side of the one bacteria, and it's not going out into the cytosol here, it's pushing it into another bacteria. It then now has two membranes around it, the neighbor bacteria and it's the bacteria that was originally in. And so it never was exposed to the outside of the cell, so the macrophages cannot get to it. So now it's into another cell, it secretes again the material that's going to break apart the membrane, it gets rid of its actin tail, now it's in the second cell, and now it's going to multiply and divide. And it's going to do that same thing over and over and over again as an infection in a person, and it's going to have so many infected cells now that the person can get very sick. So it's been hidden from the macrophages, and so your first line of defense, or one of your first lines of defense, have been avoided. So your macrophages cannot attack it and get rid of it. Okay, so as long as it stays hidden inside a cell, it's very, very hard to kill it. Yes?
How long can it remain undetected by the immune system?
I don't know. I don't really know what the effective treatment for Listeria is, because once it's hidden inside of a cell, the only thing that we can do is kill the cell itself in order to get rid of it, and then expose it so that it's on the outside and your body can actually get rid of it. But I truly don't know what the way that they get rid of Listeria. But they do. I mean, people get it all the time. Or I can't say all the time. They get really sick with it. It's like the one that gets from ice cream and it infects people and people die because it does this so fast. Before you know what happens, your body's already weakened and it dies. Or your body dies, not the Listeria. So this is just a really clever way that an organism has found to get rid of some of the immune system ability. So we're only going to cover a few more slides. These are just kind of essentials that you have to think about. So whenever you are encountering microorganisms, you have to think of how they are going to get into the body. So we already said once you open your mouth, you got bacteria coming in, it's going all through the body. If there's nothing you can latch onto, it's just going out. But there's other portals of entry, and some of those portals of entry we have to think about. So the skin can go right through the skin. Mucous membranes, placenta, parental root, and so this is like getting shots or different things, anything that's going to pierce the skin. Ears, any kind of opening that the body has. So organisms are very, very clever. Okay. And so this is just, once the organisms get into the body, then the body's contaminated, and depends if the infection becomes established, it can then cause disease. But just because it gets in the body does not mean you have disease. It has to actually cause an infection. Okay, and so just as we can get in, things will get out also. So I don't know why this won't work now. Okay. Oh. Well, we have a slide right after that one. Here he is. Okay, so the same thing here. This is just using a female for going in and a male for going out. But it's all the same places. So the idea is that organism that's in the body that wants to get out to another one, it's either you're going to have to sneeze and cough to get it out. You have to touch your mouth or something and then be able to touch something else. Come from feces, any kind of solutions that come out of the body is going to be able to carry disease with it. So these are just shows you that there's portals of entry and there's portals of exit. And for diseases, there's another list of things that are connected with how we talk about disease. So infection, when that something infects, Morbidity, this is what happens, whether it's going to kill the body or not, or kill the host, whatever it is. Etiology, this is really the disease itself. The symptoms, this is what you can tell a doctor you have. Oh, I feel sad, or I feel happy, or I don't feel good, or my foot hurts, or whatever it is. These are all just symptoms. These are subjective things that you describe about yourself. And then signs are something that a doctor can actually look at. So they can take your temperature, they can do a lot of things that are quantitative, and then this would allow them to observe it. And then syndrome, this is what they say that it's when they have a disease that has particular characteristics, we call those syndromes. So you have a chickenpox syndrome or this syndrome or something. So it's what all the groups of symptoms and signs that you have of a disease, that would be the syndrome for that disease. And so some people are asymptomatic and some people are subclinical. So you don't necessarily know you have them. The signs and symptoms don't really show up. And sometimes that's just because you're extremely healthy. Your body's already fought it off. You have it, but your body's fighting it out. And so you really never show any kind of symptoms. Okay, so these are just terms that you have to know. Again, multiple choice kind of questions. Oh, so I don't think I skipped a slide. Pretty sure I did not. No. So this is, let's go back to this. So these are also the way a disease progresses. So you have that beginning in incubation period. Sometimes if your immune system is really strong, that's it. It goes through this part and it's gone. Your immune system's kicked in, it's gotten rid of it before you even know what happened. And then you get to the point sometimes when you actually feel it. You feel tired, or you don't feel very well, or whatever pain or something occurs. And this is a point also that your immune system will be really kicking in and get rid of it before anything else happens. Or you could go to actually where you get disease. So you can't, you don't go to work, or you don't go to school, or something really happens. And that can last a long time or a very short period of time. But given enough time, your immune system usually will be able to kick in and get rid of it. If not, you usually go to a doctor and you get a little boost for that. And then those symptoms all start declining. And once they decline, this is where we make the biggest mistake. We start declining right here. We say, oh, wow, I don't have to take my antibiotics anymore because I'm feeling pretty good. I'll just save these because I know I'm going to get this again. So you save half the bottle because you think next time you get it, you'll take them on your own. So that's one of the things that happens. And sometimes when it's declining, you go right back to work. So you're still feeling pretty tired right here, feeling better than you were, but now you don't want to miss any more work because you want to save the time for vacation or whatever. But you go back to work or school or something like that, and sometimes this starts to level out, and it takes longer and longer and longer for you to get better. So a normal course of action would be that it would really have a pretty quick recovery period, and then you would be well enough to go back. But nobody hardly ever takes that. Doctor says, oh, go back in a week, and you go, oh, okay, I feel better, I'll go back in one day. But you really should always take recovery time really seriously. And these, again, there's 3 slides here in a row. I'm just going to let you kind of look at these. There's going to be nothing other than kind of the multiple choice thing. Here's a heading, and here's what it means. So again, this is just a really nomenclature-heavy chapter. And this slide also, mode of transmission, so airborne, waterborne, foodborne. I think these, if you kind of read them, they just make sense. Yep, so. And this one, there's one more that is important. These are just terms to classify them, so you can be really acute or subacute, etc. So again, things that you can kind of read on your own and just know that, again, this is a good for matching. And I'm going to stop after this one. So this really talks about epidemiology, and epidemiology when you're looking at diseases, you're trying to quantify them in some way to be able to tell the incidence and the prevalence of them. So an incident, these would be new cases, so you can see they spike up here, then they go back down again. So these would be the numbers of cases, and this will be, in every year. So they keep these records for long term to be able to see maybe some trends, maybe when the flu comes and goes, and maybe when COVID's going to come and go. So they'll keep these trends, and so they'll know how to maybe determine whether that trend is getting worse and should we do something about it or not. And then the prevalence, so these are the total number of cases that you have, and you can see the prevalence of it up here. So it will spike and then usually go back down again. But again, they can correlate this with, here's the incident happens, and then the prevalence could be even a different time frame. So these are diseases, and they record all this morbidity and mortality. They have a weekly report that all diseases are reported to, and I'm not sure that still is in effect. I think that has been done away with. So maybe some information like this that could be very valuable will not be collected, at least for a while. And the last one is just another slide, just like this. Again, these are records that are kept, and this data is the only way that we really understand what's happening. So here we have various cases of diseases by age group. Here we could have them by number of cases per month, so we know when is a heavy season for this disease or when not. And then these are maybe cases that occur in different states. So all of that kind of information, maybe just data for us, but when it's looked at over long periods of time, it really gives a good picture of what's happening, especially a small place like the United States where we can collect the data pretty easily and know exactly how many cases in the United States. Other places like Europe and stuff, it's very hard because they don't have such a coordinated health system. But still, this is always very good and valuable data. So I kind of want to close on this to make you always think that you see all this data now, and we want to kind of see in the future how much data are we still going to collect and how much data are we going to need to collect to keep the society safe. So we'll be the only ones to be able to determine that. Are we getting what we need to keep the society safe, or are we not? And if we're not, then we have to make sure that we are involved in help making changes in that. All right, so we're going to stop here, and I think there's only a few more slides at the end here. Again, some epidemiology. These are just some diseases. So nothing from the point that we just left off further will be asked. These I will not ask you. The CDC stuff I won't ask you. We might pick this up at another time about the reservoirs. So nothing that past the point that I went will I ask you on this next quiz. But if there's other stuff I think is important that you might need, we'll bring it up in another lecture. I'll just tack it onto the back of another lecture. Okay, so we're going to say this is where we stopped. Right here, after this last one. So that's where we stopped. Okay? And if you needed to look at the, and did everybody bring, if you had taken your exam with you, did you bring it back? Yeah. I don't know if I have the team now. Let me count them. I'm still missing one, so maybe I'll pick that person up in lab. Okay, and so your cards and everything, you're returning those?
Did you know? Vivint.
And if you wanted to look at the exam, I still have it, and I'm still missing one.